All right, let's dive into something truly fascinating today. Have you ever noticed how certain health conditions, like, say, heart disease or diabetes, they seem to run in your family, but maybe not in that really clear-cut way you learned about in high school biology?
Yeah, it's not like simple Mendelian traits, is it? Like eye color.
Exactly. It turns out the whole genetic story of human disease is, well, way more intricate, more dynamic, and honestly, kind of more surprising than we used to think.
Mhm.
So our mission on this deep dive is basically to journey from the absolute basics, the building blocks, to really getting a handle on why common diseases are so complex. We want to cut through the noise, the jargon, get to those aha moments.
And what's really remarkable, I think, is the shift in how we even approach this intellectually. We've moved from trying to fit everything into these neat little boxes
Right.
to embracing a much more, let's say, complex and probabilistic reality. It's a fundamental change in perspective.
I like that, probabilistic.
Yeah.
So to get started, let's make sure we're all grounded in a few key ideas. Maybe start with the term allele. What exactly is an allele?
Sure. So think of your DNA as this huge instruction manual. An allele is basically just a specific version or a variant of an instruction at a particular point in the manual.
Okay.
It could be tiny, like one letter change, that's a SNP, a single nucleotide polymorphism, we often call them SNPs. Or it could be bigger, like a whole paragraph inserted or deleted.
Got it. Different versions of an instruction.
Exactly. And you inherit two copies of almost every gene, one from each parent. So you get two alleles for each gene. That specific combination you have, that's your genotype.
You're an internal genetic blueprint.
Precisely. And then your phenotype,
Uh-huh.
well, that's the observable result. Your characteristics, like hair color, height, or even your susceptibility to a certain disease. It's the outcome influenced by your genotype and, crucially, your environment and lifestyle.
Okay, that makes sense. Genotype in, phenotype out, plus environment.
Pretty much. And if you zoom out from one person to a whole population, all the different alleles for all the genes in that group make up the gene pool.
Right.
And within that pool, we measure allele frequency. It's simply how common a specific allele is. We usually express it as a number between 0 and 1. So a frequency of, say, zero point 0.1 means that allele makes up 10% of all the copies of that gene in the population.
Okay, simple enough. So how common is this particular genetic spelling?
You got it. And, for a long time, the way we thought about genetic diseases was really dominated by those simpler Mendelian models, like cystic fibrosis, Huntington's disease.
Single gene, clear inheritance pattern.
Exactly. If you inherited the specific allele, the outcome was pretty predictable, quite deterministic. But that model, while super important, just didn't work for the common stuff. Heart disease, type 2 diabetes, obesity, most cancers, psychiatric conditions.
With things that affect millions.
Right. Their etiology, their cause, is multifactorial. It's not just one gene. It's usually many genes interacting. That's polygenic inheritance, plus significant input from environment, lifestyle, diet, you name it.
So the genes aren't causing it directly, more like nudging the odds.
That's a great way to put it. They act as probabilistic risk factors. They increase your susceptibility, sometimes by a little, sometimes by a bit more, but they don't guarantee the disease. It's a huge shift from gene equals disease to genes influence risk.
That really changes how you have to think about prevention and treatment, doesn't it?
Absolutely.
So if these alleles are being passed down and their frequencies change, what are the forces actually doing the changing? What sculpts the genetic landscape of a population over time? This gets really interesting.
Yeah, this is where population genetics and evolution really come in. We can sort of lump these forces into two buckets. You've got systematic processes, which are predictable in both how strong they are and which direction they push allele frequencies.
Okay, like a steady pressure.
Exactly. And then you have dispersive processes. These are predictable in their magnitude, like how much fluctuation they cause, but not in their direction. They introduce randomness.
Ah, okay. Predictable force versus predictable randomness.
Kind of, yeah. And the frequency of any allele you see today is basically a snapshot reflecting the history of all these forces acting on it over generations.
A living history book in our DNA, like you said. So let's break down the key players. Natural selection must be a big one.
Definitely. That's a classic systematic process. If an allele gives individuals an edge, better survival, more offspring. Those individuals tend to pass on more copies of that allele. So its frequency increases in the population over time. It drives adaptation.
Makes sense. Survival of the fittest, or maybe fittest allele.
Sort of, yeah. But then there's genetic drift. This is the main dispersive process. It's all about random fluctuations in allele frequencies due to pure chance, especially in small populations.
Why small populations particularly?
Well, imagine flipping a coin. If you flip it only 10 times, you might easily get 7 heads just by luck. But if you flip it 1,000 times, you're going to get much closer to 50-50. Drift is like that. Random sampling effects are much stronger when the sample size, the population, is small. An allele can become common or disappear entirely just by chance, even if it's slightly harmful or beneficial.
Wow, okay, so chance plays a big role. What else?
Gene flow, or migration, that's systematic. When people move between populations and have children, they bring their alleles with them. This can introduce new alleles or rapidly change the frequencies of existing ones. It tends to make populations more similar genetically.
Like stirring different pots of paint together.
Good analogy. And we absolutely have to mention mutation. It's the ultimate source of all new genetic variation. It happens slowly, constantly, creating new alleles. It's the raw material selection and drift act upon.
The ultimate source, okay.
And one more: population bottlenecks. These are events where population size crashes dramatically, maybe due to a famine, disease, or a natural disaster.
Right.
The few survivors might have, just by chance, a very different set of allele frequencies than the original larger population. It's like grabbing a random handful of marbles from a big jar. The colors in your hand might not represent the whole jar accurately. Bottlenecks are basically extreme versions of genetic drift.
So all these forces are constantly interacting, shaping the gene pool.
Constantly. And this actually helps explain something that often confuses people. Why do harmful alleles stick around? If natural selection is working, shouldn't it get rid of alleles that cause diseases like, say, cystic fibrosis?
Yeah, that's a good question. Why do they persist?
Well, it's often more complex. Take the main cystic fibrosis allele, F508. It causes a serious disease when you inherit 2 copies. But people who carry just 1 copy, heterozygotes, seem to have had some protection against diseases like cholera or typhoid fever historically.
Ah, a trade-off. Harmful in 2 copies, but maybe beneficial in 1, at least in the past.
Exactly. It's called heterozygote advantage or balancing selection. So the allele's frequency today isn't just about its current negative effects. It reflects this complex history of past selective pressures. It's a reminder that evolution doesn't have foresight.
That's fascinating. So an allele story is written by its entire history, not just its present. Is there a way to, like, read that history across the whole genome?
There is, actually. We use a tool called the allele frequency spectrum, or AFS. It's essentially a summary, a distribution, showing how many genetic variants exist at different frequency levels in the population, how many are super rare, how many are kind of rare, how many are common, and so on.
Like a genetic fingerprint of the population.
That's a great way to think about it.
Yeah.
The shape of the AFS gives us clues about the population's history, bottlenecks, expansions, migrations, and also about the long-term effects of natural selection acting on different types of mutations.
Very cool. Okay, so we have the building blocks, the forces, and ways to see their history. Let's bring it back to those common diseases. How does this whole allelic spectrum, this range of frequencies and effects, actually play into our risk for things like diabetes or Alzheimer's?
Right, so the allelic spectrum in disease refers to the complete range of genetic variants contributing to a condition. This includes how common or rare they are, their frequency, how much impact each one has, its effect size, and how likely it is to actually cause the disease if you have it, its penetrance.
And for a long time, there were sort of 2 main camps, 2 competing ideas about the spectrum for common diseases, right?
Yeah, broadly speaking, first, there was the common disease, common variant hypothesis, the CDCV idea. This suggested that common diseases are mostly driven by, well, common genetic variants. Each variant might only have a tiny effect on risk, maybe increasing it by 5% or 10%. But if you inherit a lot of these small effect common variants, your overall risk adds up.
Okay, death by 1,000 cuts, genetically speaking.
Kind of. And the idea was these variants are common because their individual effects are too small for natural selection to weed them out effectively. This model got a huge boost from the early genome-wide association studies, or GWS.
Which were good at finding common variants.
Exactly. But then there was the alternative. The common disease rare variant hypothesis, or CDRV. This proposed that common diseases might actually be caused by many different rare variants, but each of these rare variants could have a more moderate or even large effect size.
So lots of different rare mutations, maybe in the same gene or related genes, all leading to the same disease.
Precisely. This emphasizes something called allelic heterogeneity. Many different rare culprits causing the same outcome. Think about the BRCA1 and BRCA2 genes in hereditary breast cancer. There are over 1,000 known pathogenic mutations, but each specific mutation is individually very rare in the population.
So two seemingly opposed ideas. Common variants with small effects versus rare variants with bigger effects. How'd that resolve?
Well, like a lot of debates in science, it wasn't really an either situation. The reality turned out to be more of a continuum. The unified continuum model is now the widely accepted view.
Okay, tell me about that.
It basically says that the genetic variants contributing to complex diseases can span the entire spectrum of allele frequencies and effect sizes. You can have rare variants with large effects, common variants with small effects, and everything in between, sometimes even acting within the same gene or pathway, all contributing to the risk of the same disease.
So the debate was partly because the technology was catching up. GWO found the common ones first.
Exactly. GWAS was great for common variants, but it just wasn't designed to find rare ones efficiently. Then came next-generation sequencing, NGS, which let us sequence entire genomes or exomes much more cheaply and deeply.
And that's when the rare variants really started showing up.
That's when we got the hard evidence for the CDRV side of the story and could really see the full spectrum. It wasn't that one hypothesis was right and the other wrong. They were both describing different parts of a more complex whole.
Okay, that makes sense. Can we look at a couple of examples to see this continuum in action, maybe Alzheimer's?
Alzheimer's is a perfect example. The most significant genetic risk factor for the common late-onset form is the APOE4 allele. It's quite common. Frequencies range from 5% up to over 40% in different populations, but it only confers a modest increase in risk. That's a classic CDCV story.
Right.
But then you have the rare, early-onset familial forms of Alzheimer's. These are often caused by mutations in genes like APP, PSEN1, and PSEN2. These mutations are individually very rare, but they're highly penetrant, meaning if you inherit one, your chance of getting early-onset AD is extremely high.
So same disease diagnosis, Alzheimer's, but completely different genetic stories at opposite ends of the spectrum.
Exactly. One diagnosis covering very different underlying genetic causes, and type 2 diabetes is another great example, perhaps even more complex.
How so?
Well, TTD is massively polygenic. Thousands of variants likely contribute. At one extreme, you have rare, high-penetrance mutations in genes like GST, glucokinase, that cause specific monogenic forms of diabetes, basically diabetes caused by a single gene defect. These are often misdiagnosed as typical type 2 diabetes.
Okay, the rare big effect end.
Right. Then at the other end, GWS studies have found hundreds, now likely thousands, of common variants associated with TTD risk. Think loci like CDKL1, HXI, FTO. Each of these common variants typically has a very small effect on risk, maybe increasing odds by 5–20%.
The CBCV end.
Precisely. And there's likely a whole range of variants in between less common ones with moderate effects, for example. It truly spans the continuum.
So if I walk into a doctor's office and get diagnosed with type 2 diabetes, that label, it could be hiding a lot of different underlying genetic situations.
Absolutely, and that's a huge challenge clinically. Someone with a rare monogenic form might need very different management, maybe even different medications, than someone whose risk comes from the cumulative effect of hundreds or thousands of common variants plus lifestyle factors.
Wow.
It really highlights the need for more personalized medicine, doesn't it? We need to eventually move beyond just the disease label to understand the specific genetic subtype driving it in an individual.
Definitely. Okay, so we understand the complexity now. How do scientists actually find all these variants across the spectrum? And maybe more importantly for listeners, what does this all mean for actual clinical practice for our health?
Well, as we touched on, GWS was the workhorse for finding common variants. It's great for scanning the genome and finding statistical associations between common SNPs and diseases in large groups of people, but it's not good at finding rare variants.
Right, the common stuff detector.
Pretty much. For the rare variants, next-generation sequencing, NGS, has been the game changer. Techniques like whole exome sequencing, just the protein coding parts, or whole genome sequencing, allow us to directly read the DNA sequence and spot those rare changes that GWS would miss.
More direct, but maybe more data to handle.
A lot more data, which meant we needed new statistical tools. Methods like collapsing analysis were developed. The idea there is to group together all the rare variants found within a single gene and test them collectively to see if having any rare variant in that gene is associated with the disease. It boosts the statistical power.
Ah, strength in numbers, even for rare things.
Exactly. And more sophisticated methods like the combined multivariate and collapsing CMC method try to integrate information across both common and rare variants to get a more complete picture.
Okay, so the tools are getting better at seeing the whole spectrum. Now, you mentioned translating this into clinical practice. I've heard a lot about polygenic risk scores or PRS. That sounds like it could be the answer.
PRS are definitely a major focus right now. The idea is to take all those small effect variants identified by GWAS for a particular disease and combine them into a single score for an individual.
Like a total genetic risk score.
Essentially, yes. You sum up the number of risk alleles a person carries, usually weighted by how strong the effect of each allele is estimated to be from the GWAS data. The hope is that this score can predict someone's overall genetic predisposition.
That sounds incredibly powerful. You could identify high-risk people early, maybe target prevention.
That's the promise, and it's very appealing. Early identification, stratified screening, potentially very useful. But, and this is a huge but,
I sense that coming.
there's a major challenge. Poor transferability across populations.
Meaning a score developed in one group doesn't work well in another.
Exactly. And this is critical. The vast majority of GYS, and therefore the data used to build most PRS, has been done in people of European ancestry. When you try to apply these scores to people with African, Asian, Hispanic, Latino, or other ancestries, the predictive accuracy often drops dramatically.
Why does that happen? Is the underlying biology different?
The evidence suggests it's generally not because the fundamental genetic basis of the disease is radically different. It's more about those population genetics factors we discussed earlier. Differences in allele frequencies, and importantly, linkage disequilibrium, LD patterns between populations.
Can you quickly unpack LD again? Why is it so crucial here?
Sure, LD is the non-random association of alleles at different positions on a chromosome. Think of it like nearby spots in the DNA being inherited together as a block or haplotype. GWAs often picks up a signal from a common SNP that's just tagging one of these blocks. The actual causal variant might be somewhere else nearby on that block.
Okay.
But these blocks, these LD patterns, can differ significantly between populations with different ancestral histories. So a tag SNP that works perfectly well to identify a risk block in Europeans might not be linked to the same causal variant, or might not even exist on the same block structure in an African or Asian population. The PRS is built using these tags, so if the tags don't point to the same thing, the score fails.
So the score is based on markers that might mean different things in different groups, not necessarily different biology, but different genetic signposts.
Precisely. And the practical consequences are stark. We've seen PRS accuracy drop by factors of 1.6 to almost 5-fold when applied outside the discovery population. A PRS for heightened schizophrenia trained in Europeans actually incorrectly predicted Africans would be genetically shorter and have lower schizophrenia risk.
Wow, that's not just inaccurate, that's potentially harmful.
It absolutely is. PRSs for IBD, breast cancer, heart disease, they often perform poorly or inadequately in non-European groups. There was even a case where a breast cancer PRS overestimated risk in Ashkenazi Jewish women, which could lead to unnecessary anxiety and procedures.
This sounds like a massive equity issue in genomic medicine.
It is a huge equity issue. If our cutting-edge predictive tools only work well for one segment of the global population, because that's where most of the research has been done, we risk widening existing health disparities, not closing them. The promise of personalized medicine fails if it's not personalized for everyone.
So what's the path forward? More diverse research.
Absolutely. We desperately need large-scale genetic studies in diverse, multi-ethnic populations. We need to build reference data sets and develop analytical methods, including PRS approaches, that are robust and fair across all human ancestries. That's a major undertaking, but essential if genomic medicine is going to benefit everyone equitably.
Okay, let's try and synthesize this. We've journeyed from the single allele through the forces shaping populations to this complex allelic spectrum underlying common diseases. It's definitely not the simple picture many of us started with.
No, it's far richer and, frankly, more interesting. Understanding this full spectrum from rare, high-impact variants to common, low-impact ones is really the key. It's what allows us to move towards truly personalized medicine, tailoring things to an individual's specific genetic makeup, not just broad categories.
So for you listening, as you think about your own health, your family history, maybe news about genetic discoveries, remember this complexity. The genetic landscape is intricate, shaped by deep history, and unfortunately also by who has been included or excluded from research so far. And maybe a final thought to leave you with. If truly personalized medicine requires embracing this full, diverse spectrum of human genetics, what does that mean for us as a society? What steps do we need to take to ensure that powerful tools like polygenic risk scores are developed and used in a way that's equitable, accurate, and actually serves the health of everyone, everywhere?